Today we're going to be finishing Chapter 4 and talking about specialized organelles. Let's go ahead and practice a review question that might be found on an AP exam. Pause the video, read through the question carefully, and try it on your own. When you're ready to check your answer, resume this video. So let's check our answers. The surface area for cell A is about 548 square micrometers. The volume is 600 cubic micrometers. The surface area to volume ratio of cell A would then be 548 divided by 600, which would be 0.913. For cell B, the surface area is about 527 square micrometers. The volume is about 706 cubic micrometers, and the surface area to volume ratio would be 0.747. So since cell A has a larger surface area to volume ratio, it would be more efficient at transporting substances. All right, let's do another review question. Scientists assert that mitochondria and chloroplasts are endosymbionts. Name 3 pieces of evidence supporting this theory. Can you do it without checking your notes? Pause the video, try to recite your answer, and then resume. All right, let's check your answers. So this diagram shows how endosymbiosis might have occurred in the past to produce mitochondria and chloroplasts. So the 3 pieces of evidence supporting the endosymbiont theory would include a double membrane surrounding both chloroplasts and mitochondria, DNA and ribosomes found inside both of those organelles, and both of those organelles will reproduce independently from the cell. So now that we've learned about some of the major organelles that are found in every single type of cell, with the exception that chloroplasts are found mainly in plant cells and lysosomes are found mainly in animal cells, let's go ahead and talk about some of the other specialized organelles. So peroxisomes' main function is to break down fatty acids to detoxify alcohol, and as they break down fatty acids will produce hydrogen peroxide in that process. Normally, peroxide is toxic to cells, so the good thing is it's contained in this organelle. And then there are specialized enzymes such as catalase that will further break down peroxide from a harmful substance into the more harmless substances, like breaking down peroxide into water and oxygen. The cell also requires organelles to be able to maintain its structure and also help it to move. So that's the job of the cytoskeleton. As you can see in this diagram, there's some fibers that have been dyed and they fluoresce under microscopy. And so these fibers are found surrounding the cell on the outside surface and within the inside. And the main function of the cytoskeleton is for support, motility or movement, and to regulate biochemical activities. So as we zoom into the different parts of the cytoskeleton, basically there are different sized fibers for different purposes. So again, we emphasize that structure dictates function. The largest of these cytoskeleton fibers are microtubules. They're hollow, they're fairly large, and they help to maintain the cell shape. And the notable job during cell division or mitosis is these microtubules are then called spindle fibers. They attach to chromosomes and help them to separate into daughter cells. Smaller fibers are microfilaments, and they help the cell to also maintain its shape. You can find them on the inner surface of the cell membrane, and also as they contract will allow cells to be able to move as well. So in between microtubules and microfilaments are intermediate filaments, and you can see that they are involved in a variety of processes. One special area within the cell from which microtubules grow is called the centrosome. So for every cell there is one centrosome, and then as cells are getting ready to divide, this centrosome region replicate so that there's one on each pole or end of the cell. So during cell division, their microtubules come from these areas called microtubule organizing centers and form these spindle fibers that will attach to chromosomes and help them to separate properly. Now, animal cells also have structures within the centrosome region called centrioles that help to facilitate the production of these microtubules. Cells also need to be able to move and interact with their outside environments. So there are two types of organelles, cilia and flagella, that are on the surface of cells to help with cell movement or moving fluids along. So the flagella or flagellum, singular, are fairly long and they're whip-like structures. You can see the propeller-like motion on the bottom right, showing a flagellum, say on the outside surface of a bacterium. And these flagella, as they whip around, help to propel cells through the water. Cilia are found on both prokaryotic and eukaryotic cells, and they're numerous short hair-like projections, and they can help a cell to be able to move, or such as the cells lining our respiratory tract, they have cilia to be able to sweep mucus along the surface of the air passages to be able to move trapped particles from the mucus. Now let's specifically talk about plant cells. Plant cells have the large central vacuole, which contains water. They also have the chloroplasts, and the other feature that is unique to plant cells is the cell wall. So cell membranes help to regulate the movement of materials in and out of the cell, but the cell wall's main job is for support, to protect the plant, and to maintain the shape. It's made up of a material that's called cellulose, which we actually, as humans, cannot digest, but the bacteria that are in our gut can produce the enzymes to be able to break down cellulose. So when we eat plant parts that have cellulose, we often call that a fiber diet, and that's actually good for our digestive health. Now, there are channels between plant cells that allow molecules to move from cell to cell, and these channels are called plasmodesmata. So you can see them in the pictures on this slide. Animal cells do not have a cell wall, but the rigid structural support for animal cells is called the extracellular matrix, or the ECM, and it's located outside of the plasma membrane. On the inside surface or intracellular portion of the cell, we see the microfilaments there helping to maintain cell shape, and then on the extracellular matrix, it's composed of a variety of different glycoproteins such as collagen, and it forms a strong network of fibers to be able to strengthen tissues and allow the transmission of external signals to the cell. So you can see that some of these proteins are actually connected from the cell membrane to the extracellular matrix for anchoring and for being able to communicate signals outside of the cell. In addition to the extracellular matrix, animal cells have also a variety of intercellular junctions. There are tight junctions that form a quilting-like effect to anchor two cells together to form a watertight seal. There are desmosomes that you can see in the picture there. They are rivets that fasten adjacent cells into strong sheets. So you can kind of see they're like the rivets that might hold different layers of jean pants material together. And then there's also channels that allow materials to cross from animal to animal cell, and those are called gap junctions. And so ions, sugars, and small molecules can pass through. So these channels between plant cells are called plasmodesmata, but between animal cells, they're called gap junctions. All right, so finally, let's summarize differences between plant and animal cells. Plant cells have that central vacuole, chloroplasts. They have cell walls, which are made of cellulose, and those connections, those channels called plasmodesmata. Animal cells have lysosomes, centrioles within the centrosome region. They do have flagella and cilia, and they have the connections between the cells to hold them together are desmosomes, tight junctions, and gap junctions. And then on the outside surface is the extracellular matrix. All right, the final review question. See if you can answer this without checking your notes. But can you list 3 differences between plant and animal cells? If you can answer this question, you've done a good job listening to this lecture and learning about the similarities and differences between plant and animal cells.